Morning, everyone. Welcome to Grand Rounds. We got the photos out of the way, but there's going to be more photos when we give the plaque, and then when the lecture's over. Everyone is associated with the Sackler Institute. In one way or another, please stay, because we're going to get a photo of everybody at that time also. So I guess this is by way of saying this is a special day. We have an award to give. We have a special lecturer who's actually going to give 2 lectures here and then this afternoon at Cornell. And we have a ribbon cutting of some new laboratories, and we have a celebratory event. Not everybody's invited to everything, but this is just part of a busy schedule, so the awardee will need to pace himself, but I'm sure he's already learned that. Just want to make a couple of announcements before introducing Jay Gingrich to preside. 3 of our residents won APA awards that we were notified of this week. Dan Kimball won a leadership award. Terry Ann Nicholson won a diversity leadership award. And Sarah Kaufman received a public psychiatry fellowship. And Deborah Cabanis received 2 awards, but I mean, she's got so many of these things that it's kind of getting redundant to mention it every time. But this one from Castle Connolly, and another one from the American Psychiatric Association for her innovative teaching of psychotherapy. Well, as I said, this is really a special occasion for this Grand Rounds today, as it's in honor of the amazing work that the late Dr. Mortimer Sackler and his family, through their foundation and various supported institutes, have conducted these past couple of decades. And to introduce that and the speaker today, I want to ask Jay Gingrich to come forward. Jay?
Thank you, Jeff. It's a real pleasure for me to be able to give this introduction as director of the Sackler Institute. It comes with a few privileges. I want to welcome all of the family members who are here from the Sackler family, as well as we have the heads of a number of the other Sackler Institutes in attendance, and we're going to be participating in our first International Sackler Scientific Summit tomorrow all day. So it is a busy period. But before we got started, I wanted to say a few words about Mortimer. He really is a remarkable man, and what I wanted to do, let me see, is this the way I advance? The one thing I've always realized is, being a psychiatrist, is you always have to think about remarkable individuals in the context of their family. And Mortimer was one of 3 brothers. All 3 brothers went on to be psychiatrists. And what makes it even more interesting for me is that, well, Mortimer was a middle child, as am I, of 3 boys, but I don't want to make any grandiose interpretations. But this is an old photo. It's the only one I could find of Mortimer with his older brother Arthur. I couldn't find one with his younger brother Raymond, so it's not a slight. Actually, Kathy Sackler here, shown in 1974 with her father, and Elizabeth is the daughter of Arthur. This is at some opening at the Smithsonian in 1974. But together, back in 1950, shortly after they had finished their residency training in psychiatry, they went to Creedmoor out in Queens and founded one of the earliest, if not the first, Institute for Psychobiological Studies. They were really interested in identifying the biological underpinnings of mental disorders. So in that sense, they were really well ahead of their time. And in fact, they influenced a generation of psychiatrists. Many of you may not know that Donald Klein actually trained both at Creedmoor and at the Institute. And who became a giant in the field. But later, together they partnered in a novel pharmaceutical venture. And in that they prospered, and it was based on that they were able to be such generous philanthropists. I'm focusing now on only what Mortimer has done, but really the entire Sackler family is a legacy of generosity which is difficult to capture in any short period of time. But of relevance here is that Mortimer and his foundation went on to found 7 institutes for brain development. The original one was at Weill Cornell. A few years later, our institute was founded, and then subsequently several institutes were founded both in the UK as well as Canada. And the prize was established near the end of Mortimer's life in 2008 by his family to commemorate his legacy of both scientific achievement and his interest in biologic underpinnings of development. And this was what's stated in the actual intent of the prize, is that it's intended to recognize outstanding achievement in the field of developmental psychobiology, including the advancement of our understanding of the developmental processes of mind, brain, and behavior that contribute to normal development and the origins of mental illness. The prize is currently awarded biannually, carries a nice stipend, and the award is voted on by a pretty large committee that's comprised mainly of the heads of the various institutes, as well as some independent individuals outside the institute. And so arriving at a consensus is something I think all of our winners should feel quite happy about. And prior recipients who've contributed to our understanding of normal and abnormal early development came from a diverse series of fields. Our first winner was Avshalom Caspi in 2008 for his work linking genetic variants in the serotonergic system with environmental adversity and the way those combined to actually generate unexpected psychiatric outcomes. Our second recipient was Fernando Nottebohm, who was recognized for his work linking birdsong learning of their actual songs and adult neurogenesis, something which has become a rapidly growing field in terms of adult neurogenesis and its role in cognition in mammalian species. In 2012, we awarded this prize to Carla Schatz, largely for her discovery that the spontaneous neural activity of neurons in areas of the visual cortex are crucial to the development of circuit structure that's going to persist later in life. Very seminal, very influential finding. And in 2014, our most recent recipient was Huda Zagbi, recognizing her discoveries regarding MECP2 and Rett syndrome, among any number of other contributions, but really all focusing on the genetic and epigenetic mechanisms that are required for proper brain development. And again, another very worthy recipient. And then this year, we have, as Jeff alluded to, Takao Hensch. And I think the prize committee was really swayed by his pioneering work in the area of critical period plasticity. That is, how do critical periods emerge? How do they close? And how might they be reopened for later remediation? Something of critical importance when we're thinking about neurodevelopmental disorders. And as he's going to talk about today, especially his work looking at Parvalbumin GABAergic neurons in this process, as well as the perineural nets that appear to be critical. I'm not going to go too extensively into his biography. It's quite notable for his many achievements, his productivity in high-profile journals. I think what's hard to capture in a CV is the influence that Takao's work has had on any number of other individuals around the world. But he does have some interesting facets. He was one of the early founders of the Riken Institute in Japan. He's gotten a number of, he was the only person I've known that got both the Young Investigator Award, both from Society of Neuroscience and the equivalent in Japan, and a few years apart. He also was an early awardee of a Pioneer Award from NIMH. And he's been an editor on many journals, including J Neuroscience and Neuron. I'm very aware of this because he's been having to hound me a few times to get my reviews done. With that, I think I'm not going to go further into his biography because I don't want to eat in too much to his time. But I do want to invite Takao to come up, and we're going to give you your check, your plaque. If I can invite Jeff and Bill, and Kathy and Susan, do you want to come up for this? So we're presenting you with the Mortimer D. Sackler MD Prize. Yeah. I'm done.
Well, thank you, Jay and the Sackler. This is a tremendous honor for me, totally unexpected, and I'm just very much honored to be in the company of those past recipients and a part of your broader family now. I've been fortunate, especially at Columbia over the past few years. My friends here have invited me every spring, it seems, and that's very nice because I was hoping on this busy day to take this opportunity to talk about some newer things, unpublished things, since many of you have heard me give talks in the past few years, but to capture the flavor of the science that we do and why we're so interested in these structures that were just mentioned. But I'm hoping that it will seed discussion and generate new ideas. And our interest is very much translationally motivated. So while we do our work at a very fundamental basic neuroscience level, we're hoping to provide new insights and new ways of thinking about mental illness and development gone awry. Our issue of interest is this very simple, almost anecdotal observation that the infant or child brain is particularly malleable. We've all experienced this. And we'd like to understand why that is. Why is the young brain so vulnerable but also sensitive to opportunity? And that these structures somehow seem to consolidate and become harder to change, in some cases irretrievably. And there must be some biological reason for this, as well as an evolutionary reason for this. And we turn often to the mental illness literature because we find repeatedly evidence that most, if not all, mental illnesses seem to have developmental origins, which is actually the theme of our Conti Center up at Harvard. And just to borrow a slide from Francis Lee, who's here, we all have seen pictures like this, where the emergence of these top 10 mental illnesses seem to happen during particular windows in time, which is not to say that certain events earlier or later are not important, but this is particularly striking. And so it's our mission in the lab to try and understand how these windows come about. And in fact, these are just a reflection of what the brain is naturally trying to do, to adapt to the environment and conditions into which the human or the animal is born. And so we talk about critical periods, and I'm not going to parse critical or sensitive periods here. I'm just going to refer to them as critical periods so you know what I'm talking about in terms of developmental time. And the idea here is that we are all born with circuits that are laid out beautifully through gene programs, hopefully, that run well and establish the basic neural networks that we can then layer on top experiences. And that this interaction, the famous gene by environment interaction, has an opportunity to reshape our circuitry during these critical periods. And once these windows close to a greater or lesser extent, our brain functions are largely set in a particular direction and shape our behavior as adults. And so, of course, there's not just one critical period. There are multiple such windows, and they are sequential in nature, such that primary sensory areas might be shaped earliest. These are the first filters of the sensory input to our brain and need to be established early on, which then segue into sensory motor integration and higher order cognitive functions. And one can imagine that timing is crucial because you can't build higher order function with a rocky foundation. And so, in fact, we use these primary sensory areas as a good workhorse model to try and understand what are the factors that turn on, determine the duration, and turn off these kinds of windows, and use those kinds of signatures to map other critical periods, increasingly in other brain regions or in disease models, for example. And so, as I showed in the previous slide, a common theme in our lab is this idea of balance, balancing plasticity with stability. And underlying all of that is a balance of excitation and inhibition. In fact, in cortical areas where we focus most of our attention, we know that the majority of neurons are excitatory. They have this lovely pyramidal shape with dendrites extending northward and axons south, and the dendrites are studded with dendritic spines, which are the focus of so many research laboratories for cellular and molecular mechanisms of synaptic plasticity. But several years ago now, we found that it's perhaps not just this population that's crucial for determining critical periods. In fact, the surrounding minority population, the inhibitory cells, which use GABA instead of glutamate as a neurotransmitter, are particularly pivotal in determining when these plastic windows come about. Inhibition has been a very interesting topic. Already 100 years ago, Ramon y Cajal had identified this rich heterogeneity of morphologies with these colorful names describing their shape and connectivity. And so it was quite a challenge to try and understand which of these cells might be important in developmental plasticity. But I will give you an overview and then some very detailed new studies that focus on one particular class, this basket cell inhibitory neuron, so-called because, as you'll see in a few minutes, they make these lateral inhibitory connections and form pericellular synapses around the cell bodies in a kind of basket or nest-like fashion. And it's the maturation of these cells which we feel is most pivotal for turning on these critical periods. And as a result, we have a cellular signature that we can trace across brain regions and try to identify whether a brain region is in more or less of a plastic state. So this is what I was just mentioning, the basket cells, also known as Parvalbumin cells in this case, because the ones we're interested in contain a calcium binding protein called Parvalbumin, or PV for short. There are, of course, other Parvalbumin positive cells, but they're not basket cells. And these Parvalbumin basket cells form these nest-like synapses around the cell body. This is Cajal's drawing. And now, fast forward to the modern day, we can actually trace these baskets using state-of-the-art techniques such as rainbow viruses, coloring all of these terminals with a different fluorescent protein, and allow us to visualize in great resolution the formation and malleability of these basket-type synapses. And so we can finally address questions such as, what is a basket synapse exactly? Is it the proliferation of one axon that forms many boutons, or as you can see here, the convergence of many different colors to form a basket-type connection? And using such techniques, we can trace when these cells and synapses mature in normal development in different brain regions, as well as in mouse models carrying genes that are linked to mental illnesses. Another interesting feature of these cells is that as they mature, they themselves acquire an extracellular coating, which we call the perineuronal net. And as these critical periods close, these cells get a tight wrapping around their cell bodies. And here's a small video of such a, I hope it works. Oh, it's not working, but you can still see. The purple here is a label for this extracellular matrix structure called the perineuronal net, and it's perforated by synaptic boutons, in this case, of other inhibitory neurons impinging on this cell body. We can use modern techniques like super resolution imaging, such as storm microscopy, to quantify these perineuronal nets now in great detail. Basically, we take these three-dimensional structures, imagine the cell body and its proximal neurites wrapped in this kind of glove, and then unfold this 3D structure like you would a globe onto a two-dimensional space, and then you can quantify and see that compared to young visual cortex, in this case, the more mature visual cortex shows a very tight wrapping, and the associated holes where the synaptic inputs would be are changing in shape. And it's quite a dynamic structure, which is not done justice by these static images. And so by using super resolution techniques, we can start to quantify the development of these structures which signal the closure of critical periods, not just in the visual cortex, but across brain regions. And these cells are interesting because they mature at different rates in different brain regions, consistent with differential timing of critical periods emerging in different brain regions at different times. And so there's a lot of evidence mounting that this one cell type in particular might be an interesting biomarker, but is it really involved in a causal manner? Well, years ago, we had shown that using this simple model of monocular vision and the loss of visual acuity in the visual cortex associated with a strategically timed patching of one eye, you can map out a critical period. And in fact, if inhibition is delayed in its maturation, either by direct genetic tampering with these cells or GABA release, for example, or environmentally, in this case, in the visual domain, by raising animals in complete darkness, these inhibitory circuits mature more slowly. This window of time becomes divorced from the chronological age of the animal and can be shifted into adulthood. And so in this case, for example, if you raise animals in darkness into adulthood, they retain the ability to become plastic as soon as they're brought into the light. Conversely, we can deceive the system by artificially enhancing inhibition prematurely. One early example was to use a drug like benzodiazepine drugs. Commonly used sedative or anxiolytic drugs like this, in fact, will cause the weak inhibition prior to this critical period to be boosted. And in fact, the system is entirely capable of plastic change at this young age. It simply wasn't being tapped properly. And this kind of result gave us a bidirectional control over when these windows might occur. Over the years, this has been replicated many times, not just by ourselves, but several laboratories. And I don't expect you to read all of the molecules and manipulations. If you just focus on the central column, suffice it to say that we now have a variety of animal models in which this window of time has been shifted earlier or later. And these manipulations are all focusing on the maturation of these inhibitory circuits, the Parvalbumin positive basket cell in particular. The duration of the critical period can be narrowed or lengthened. These manipulations are related to the actual dendritic spine maturation and plasticity on the excitatory neurons. And critical period closure has also been manipulated, and I'll talk more about this a little later as well as this evening. The ability to open a critical period but never close it might seem attractive if you want to stay plastic throughout life, but there are probably also consequences for doing something like that. So I think the last time I was here, I picked one example, is there a clock that determines the timing of these windows? And I won't go through that story in detail. I'll just say that we can address these kinds of questions by manipulating molecular players that are involved in clocks in the brain. And if you disrupt clock signaling in these inhibitory neurons in particular, you get this effect, that the normal critical period timing is shifted later or delayed. In fact, these cells develop more slowly in the absence of their clock gene. And this might have implications for why critical periods happen at particular times in particular brain regions. These clocks may be running at different speeds, they may be engaged in different fashion, depending on when sensory experience kicks in. It also has implications for the use of embryonic inhibitory neurons as a kind of stem cell therapy. This has been attempted out in the West Coast by Alvarez Buya and others there. to scoop out some of the medial ganglionic eminence, the source of these inhibitory neurons from the embryo, and transplant them into the postnatal visual cortex, for example. This, in fact, does recreate a second critical period, but only when the transplant matures about a month or so, suggesting that there is a timer intrinsic to this system that determines when these windows turn on. From a clinical point of view, many of you are familiar with the fact that circadian genes and SNPs in these genes have been linked to a variety of mental illnesses, and typically the impact of circadian genes on cognition is interpreted as an acute effect of disrupting the circadian rhythm. We've all experienced this with jet lag. We know that playing with the circadian rhythm is detrimental to cognition, but our results offer another possibility, that in fact people who carry these polymorphisms may have also incurred a delay in developmental trajectory that should have been happening at the right time, at the right place. So this is one way in which a kind of bottom-up view of mechanisms that determine critical periods might start to inform other much broader picture questions. And what I wanted to do today was focus on the role of inhibition in turning on or executing this kind of plasticity. This is a kind of vexing problem because it seems kind of paradoxical that you need inhibition to allow brain plasticity to turn on. And in early studies, in collaboration with Ken Miller, who's here, we analyzed some of our recordings and found that there may be one role for inhibition, and that is to sharpen the signal to noise. That is, in the case where inhibition is still weak, we find that there is still quite a bit of spontaneous activity that can be recorded, and this could be detrimental to classical spike timing dependent plasticity rules such as fire together, wire together, or out of sync, lose a link, these kinds of mantras in our field. With the maturation of inhibition, the noise is preferentially dampened and the evoked visual responses become more crisp, and these rules can be engaged more effectively. So one idea from this sort of modeling work, as backed up by experimental results, is that inhibition is necessary for coding these kinds of plasticity rules to be engaged. But we felt that there was something perhaps more pivotal about these cells. And the perineuronal net, as I've been alluding to, gave us some suggestion that it might be plasticity onto these neurons themselves, which is particularly interesting. In fact, if you take this artist's rendition, I said the net is basically a glove around the cell body of these inhibitory cells perforated by synaptic boutons. And as the glove gets tighter, as you saw in the storm images, it may become physically obstructed and difficult to rewire these synapses onto these inhibitory cells. Let's see if this works. No, my movie doesn't work. But the right side would simply be a real version of this cartoon. And so the question became, are these inhibitory neurons themselves plastic? And in fact, we now know that they are. Yoko Yazaki in the lab, originally in mice, and here a replication of our results in the cat by Marcos Frank's group, showed that, in fact, the fast-spiking inhibitory interneuron, this Parvalbumin positive basket cell, is actually quite plastic. They normally respond, in the case of the visual system, equally well to either eye input, but when you patch an eye to induce plasticity, these are the cells which respond first, and weirdly so, because they shift in favor of the closed eye and then in favor of the open eye later. And only later do the excitatory neurons follow suit. Again, with more modern techniques, you can visualize the parvalbumin cells selectively with fluorescent proteins, as Josh Trachtenberg did here. And if you patch onto these cells specifically, as opposed to relying on their fast-spiking electrophysiological signature, you see the same thing, that within 1 day of closing an eye, these cells lose half their input. And so it seems that these neurons are pivotal not only because they can clean up the noise on the excitatory cells that receive their inhibition, but they themselves might be plastic, and they might be plastic first. in order to identify how fast is this plasticity, we developed a thalamocortical brain slice preparation, which retains the connection from the visual thalamus to the visual cortex. And using voltage-sensitive dye imaging, for example, you could stimulate the lateral geniculate nucleus and see a response in the visual cortex. And using this preparation, Katie Quast in the lab recorded the strength of input from the visual thalamus to the visual cortex, and found that, as reported for other sensory systems like the barrel field or the auditory system, these inhibitory cells in fact receive an overwhelmingly stronger input from the thalamus than their neighboring excitatory neurons do. And this is already mature before the critical period even begins. And so what happens when you patch an eye? Well, as Delma Coyote showed in the lab, they lose 50% of their input immediately. Within 2 hours of monocular deprivation, the thalamic input to these inhibitory neurons is cut in half. These changes are not seen in the excitatory neurons. And in fact, it's directly relevant to the critical period, because if the monocular patching is done before the critical period, or in a GABA deficient mouse that fails to enter the critical period, these changes are not seen. And a variety of measures, both anatomical and physiological, suggest that this is a presynaptic change of thalamic input onto these inhibitory neurons. So if it's really this fast, we should be able to read it out also at a systems level in a very fast manner. And so Rebecca Ray, another graduate student in the lab, started to focus on electroencephalographic signatures, EEG recordings from the visual cortex of these young mice. Another key function of these parvalbumin positive basket cells is that they generate oscillations. And a very famous oscillation that you're familiar with is the gamma rhythm. 30 to 80 hertz oscillations in the EEG are typically driven by parvalbumin positive inhibitory interneurons. And they are this fast desynchronized state that's typically associated with perception and cognition and compromised in many mental illnesses. What Rebecca found was that compared to non-deprived animals, the frequency, the power in this frequency range shows a transient peak around 42 hertz, in fact, 1 hour after monocular deprivation. And if you look at this time plot, the first 4 hours after deprivation, you see that there is this peak in the gamma range. It shifts slightly over time in the first few hours. And those with a sharp eye might see that there are these blips of yellow, which are reactivation of this gamma during rapid eye movement sleep. And so immediately after patching an eye, we see a loss of thalamic input to these inhibitory neurons and a clear signature of altered excitatory inhibitory balance onto these cells, a rise in gamma power, which then eventually goes away. So this was quite intriguing, and Rebecca tested a variety of conditions. For example, is it specific to the visual cortex if you're doing a visual manipulation? And in fact it is. There's no change in the somatosensory cortex. It's dependent on visual experience after patching the eye, because if you put the animals into a dark box after patching an eye, there is no prominent peak like this. And so the experience of the animal seems to drive a very rapid change in gamma power. We turn then to our favorite reversible critical period model, a GABA-compromised mouse in which GABA synthesis is turned down through the loss of GAD65. These animals normally don't show a prominent gamma peak, but we can rescue plasticity and turn on the critical period using benzodiazepines, as I mentioned before. And in fact, when you do that, monocular deprivation now produces a gamma peak. And in fact, this can wash out. And so you see that this gamma seems to be a kind of biomarker or signature of whether the brain is in a critical period state or not. If we go to the adult animal, where we know that patching an eye does not produce this kind of visual acuity loss or plastic change, there is no gamma peak induction. But if you take a dark-reared animal as an adult, you get a beautiful gamma peak. Another brake-like factor which turns off plasticity, I'll talk more about this evening, is the LINX1 molecule. Mice lacking LINX1 also show a deprivation-induced gamma peak as adults, and that's shown here in their surf plot. So again, you see this elevation in gamma and some repeated replay during bouts of REM sleep, and it only lasts a couple of hours after deprivation. So of course, these days, graduate students aren't satisfied with a thesis if they don't use optogenetics. And so of course, this had to be done. And so parvalbumin positive cells can be loaded with channel rhodopsin. And if you flash light at 40 hertz to simulate gamma rhythm, you can generate a beautiful peak in the power spectrum artificially. And Rebecca paired this transient 3 hours of gamma with monocular deprivation in adult animals and found that, in fact, you can induce visual acuity loss in adults in this way, and that, in fact, the gamma for just a few hours at the onset of deprivation might be priming the pump, allowing something to happen for plastic change. And so we started to dig deeper into this phenomenon. How is gamma generated? Well, there are a number of ways that involve parvalbumin cells. For example, the famous one is local circuit inhibition. Parvalbumin positive inhibitory neurons inhibit each other through chemical synapses, but they're also coupled by gap junctions, and this creates a kind of synchrony detector in this network, and ultimately they inhibit the neighboring excitatory neurons with those famous basket synapses. And this kind of ping or ing model, depending on what which perspective you have, can generate in computational models very beautiful gamma oscillations. Another way to drive gamma, Bob McCarley has shown, is from parvalbumin cells in the basal forebrain. So these are long-range axons that are inhibitory, containing parvalbumin, which project broadly through the cortical mantle. And we decided we would test this model experimentally by disrupting the inhibitory receptors made between parvalbumin cells. So if we took out this crucial synaptic component, could we have an impact on gamma oscillations? But it wouldn't be enough to damage the system. We would like to restore the balance again and try to rescue any kind of disruption. And so we turn to metabotropic glutamate receptor, which some of you might be familiar with, the mGlu2/3 receptor. There are selective ligands for these receptors. And as you can see by fact-sorted qPCR here on the right, as well as microdissection of visual cortex versus basal forebrain, mGlu2/3 is highly enriched in the visual cortex, but not in the parvalbumin cells. It's found in the excitatory cells. And so, in fact, if you were to record from a parvalbumin positive cell, the excitatory postsynaptic currents can be dampened by such a drug, an mGlu2/3 presynaptic receptor agonist, whereas the inhibitory postsynaptic currents are not affected. And so, we have a genetic model in which the EI balance is disrupted onto the parvalbumin cells by removing their specific alpha-1 receptor, and potentially a pharmacological tool to further dampen excitation selectively onto these cells and maybe restore the EI balance back to what it was. This is a remarkably clean manipulation. Total knockout of the alpha-1 subunit leads to massive compensation by other GABA-A receptor subunits. But if you just remove alpha-1 from the parvalbumin cells, which are long known to be enriched in this subunit, you just get rid of that. And there's no change in any of the other GABA receptor subunits in the parvalbumin cells or in the non-parvalbumin cells, nor is there any change in the connexin or in the gap junction protein or in parvalbumin itself. Physiologically, the effects are very specific. If you record miniature inhibitory postsynaptic currents from parvalbumin cells, you see that they are smaller and fewer in number, and the ones that remain have this long, slow decay characteristic of alpha-3 containing GABA receptors, non-alpha-1. And pyramidal neurons instead show no effects of this cell-specific deletion. Parvalbumin, by the way, is slowly developing protein, and so the use of this creline relegates the excision to late postnatal stages, so we avoid any kind of issues with early deletion. So now you've been dying to know what happens. Gamma oscillations are in fact enhanced in these animals, and so across brain regions we find that between 40 and 80 hertz there is this very prominent bump in gamma power. You can see it by eye in the EEG trace, and if we inject these animals with the mGlu2/3 agonist, we can rescue that and restore gamma power to more normal levels. So we have a reversible gamma switch. These animals are running around with heightened gamma all the time, but we can turn it down when we want to. This is perhaps more visually appreciated here. This is frequency from 0 to 80 hertz across time, and in wild-type mice, there's not so much gamma. You inject the LY drug, and not much changes. But in the knockout mouse, they have heightened gamma, and boom, with this drug, it's switched off for several hours. And so we use this trick to test what the impact is on adult plasticity. As Rebecca's optogenetic experiment suggested, if you have heightened gamma, that might be priming the pump for plasticity. And sure enough, these animals have extended into adulthood a sensitivity to monocular deprivation, shown here in red. Wild-type mice at this age and well beyond, we've done this up to postnatal day 200, show no loss of acuity with deprivation, as shown here in the actual data. But these knockout mice retain the ability to change. But as soon as you turn off the gamma with this LY manipulation, the plasticity is lost. Yes.
Can I ask you a question?
Yeah.
Go back to the last slide.
Yes.
So, if the P3 agonist rescues
Yes.
these suppression to gamma oscillation,
Mhm.
presumably it's doing so by enhancing the inhibitory neurons.
It's by dampening the excitatory transmission onto the interneurons.
So
Yeah.
what happens if you administer
We haven't done that in this mouse, but we're welcome to try all of these manipulations. The logic was, can we offset the postsynaptic inhibitory weakening with some dampening of presynaptic input? And of course, this drug has been sort of en route to clinical trial at one point, so it has some merits that we might get into. We also tried a different critical period paradigm that I'll talk about this evening that probes the prefrontal cortex. This is a preference behavior. Animals will choose to form a nest given the opportunity in these little shelters. Typically, they prefer the silent shelter, but if they're plastic, exposing animals to music will make them prefer the music chamber. And the knockout mice, in fact, start out normal, but when exposed to music for 2 weeks, they switch their allegiance from silence to music, whereas the wild-type mice don't. That's shown here in terms of the cumulative fraction of mice that prefer music. But if you dampen the gamma during this music exposure, they don't show that plastic change. This is a little faint, but so we can switch it on and off. This is where things became interesting. We were wondering, what is it about gamma that makes the brain plastic? And this is where the three components of my title come together, because the perineuronal nets turn out to be a very sensitive measure of the gamma. In fact, this complex structure can be quantified. Here we drawn by hand the coverage of a perineuronal net around the parvalbumin cell in wild-type adult mice. Here it is in the knockout, clearly compromised in structure, and the knockout treated with the LY drug, the net has recovered, and that's all quantified here in terms of branch length or branch number. We also looked at a panel of approximately 25 genes which contribute to the structure and breakdown and turnover of perineuronal nets, and a handful of these show increased expression in the PV alpha-1 knockout mouse, consistent with rapid turnover, and most of those are restored to more normal levels by this LY treatment that dampens the gamma oscillation. And so how does this perineuronal net turnover come about? We think it's because of mild oxidative stress. With Kim Do's laboratory in Lausanne, we had shown that the perineuronal net has, among one of its many functions, the ability to buffer these cells from their own oxidative damage. And in fact, oxidative stress is a very well-studied cell biological process. We know that as oxidative redox imbalance builds, the synthesis of glutathione shunts the transsulfuration pathway, and so you can measure the thiols in the brain and quantify, is there some kind of signature of oxidative damage in this high gamma situation. And just to illustrate here, you can see that cystathionine, an intermediary from the reverse transsulfuration pathway to production of glutathione, is in fact elevated. The oxidized form of glutathione, the natural antioxidant, builds, and therefore the ratio of glutathione to oxidized forms goes down. All of these features of oxidative stress are reversed by dampening gamma oscillations with the LY drug. And so gamma may be producing a mild form of oxidative damage, which causes the perineuronal net to disassemble. And in fact, we can see that this is true, especially in the parvalbumin cells, using a different marker of oxidative damage, mitochondrial DNA damage marker. You see that this signature in the weakened net positive cells is in fact enhanced. And so we think that we have an interesting possibility that gamma, which is a prominent component of parvalbumin cell activity, may actually allow plasticity if it goes on long enough, because it's loosening the net around these cells. It's also interesting from a clinical point of view, because in a perspective piece we had a small part contributing to with Kim Do, it seems that oxidative stress is a hallmark of almost all, if not all, models of schizophrenia, if I can take the liberty of calling it that. Here are a variety of environmental or genetic models, and in all of these cases, there are signatures of oxidative stress and parvalbumin cell impairment. And as to perineuronal nets in particular, we know from the work of Sabina Beretta up at McLean that in postmortem brain tissue from patients, the perineuronal nets in prefrontal cortex are reduced in density and complexity. And so last time I probably showed you a slide like this, that one feature of critical period closure might be to protect these cells and the brain from oxidative damage, and that one way to produce this kind of oxidative damage might be heightened, at least transiently heightened gamma oscillations. I was very excited to hear from Peter Uhlhaas, who has been studying high-risk subjects for schizophrenia and comparing with MEG measures of gamma, baseline gamma, just like we've been recording in our mice, their gamma versus controls, and first episode patients versus controls and chronic. And he's finding that, in fact, there is in high-risk subjects before their first episode heightened gamma, which then transits to this well-known pattern of reduced gamma as you go into the chronic stages. So what is this perineuronal net doing at a molecular level to keep these cells healthy? We know that they're composed of sugar-bearing proteins, chondroitin sulfate proteoglycans, these purple wavy molecules here. And one function might be to attract a whole host of molecular factors which these cells need for mature, fast-spiking behavior. And what's interesting about these molecules is that none of them are made by the cell, the parvalbumin cells themselves. Although they're highly dependent on it, on them, they are getting it from their local cellular milieu. And many of them are activity dependent. And so, in fact, the delay in parvalbumin cell maturation in a dark room is in part due to the lack of availability of these kinds of factors, say BDNF, which is secreted in an activity-dependent manner from nearby cells. We study one molecule in particular, OTX2, which you see here nicely nestled inside these perineuronal net-bearing cells. But this molecule is even more bizarre. It's not even made in the cortex. It's made quite far away and somehow has to find its way into these parvalbumin cells. The way it does so is at a molecular level, there is a glycosaminoglycan binding motif in the OTX2 protein, and this short peptide sequence can in fact bind the molecules to these perineuronal nets and then be internalized into these cells. The motif can be used as a small peptide antagonist. And so if you inject into the adult brain this small peptide called RK peptide, you can deplete all the yellow cells which show co-localization of parvalbumin with OTX2 of their endogenous OTX2. Physiologically, this turns these highly fast spiking cells into somewhat less fast spiking situation. So compared to the mature fast spiking properties, they are shifted down to a less fast spiking state. And in fact, under these conditions, visual cortical plasticity anyway was knocked back into a critical period state, and you could rescue amblyopia in adult animals. We know, however, that OTX2 is not just relevant for the visual system. It's in fact synthesized in the choroid plexus. And this motif, when mutated in a mouse line, so the RK component of that peptide, if you change it to a double alanine in a knock-in line of mice, shows a delay of critical period plasticity, not only in the visual cortex, but throughout the brain, in the auditory cortex, in the prefrontal cortex. And it suggests that this source might be a kind of master regulator, and its strategic location in the choroid plexus is particularly appealing, again, from a clinical point of view. This is a leaky part of the blood-brain barrier. If you could address a drug that would regulate the level of OTX2 in the choroid plexus, you might be able to rescue the development or developmental mistiming in animals. So I'm going to end with one example where we've used this strategy. Is there a condition where parvalbumin circuitry is, for example, too mature, that this might be a problem worth treating? A number of years ago with Michela Fagiolini, who's here in the audience, we've been studying the MECP2 knockout mouse model of Rett syndrome, and since Huda Zogby was the last recipient of this award, I felt it was appropriate to talk about this work. In 2012, we show in this paper that MECP2 knockout mice have a clearly elevated connectivity of parvalbumin positive circuits. Ariana Maffei and Josh Wang have since shown that, as predicted, this accelerated maturation in fact leads to an accelerated critical period timing, so all good there. And also in human tissue, we find that these inhibitory cells are pumped up. But like all other features in the Rett syndrome animal model, vision, in this case, develops and regresses. And so Michela has really turned this into a very wonderful biomarker to track the condition of not only the mouse, which shows this classic hind limb clasping and a variety of Rett phenotypic scores, loss of visual acuity, but also in people. And so with Chuck Nelson, she published this work showing that visual acuity in Rett syndrome girls is in fact decreasing. And more so, you can even use the visually evoked potential waveform here in black, the typical wild type condition, and see that as regression proceeds in the mouse or in the human, the waveform gets broader and smaller, and we can actually diagnose or predict the severity of the mutation or the severity of the regression simply by a few minutes of visually evoked potential recording from the surface of their scalp. A very powerful tool indeed for tracking patients who lose their language and ability to convey their symptoms. Well, in fact, it's not just in the visual cortex. At a glance, you can see that parvalbumin signal is elevated everywhere throughout the brain compared to wild-type mice. And in postmortem human tissue, it's particularly prominent in the middle layers of cortex and correlates with an elevation of OTX2 in the cerebrospinal fluid of these Rett patients. Here, for comparison, we look at neurotypical subjects, CSF, not Rett syndrome, those with meningitis, a well-known compromising factor to the choroid plexus, and those without. And you see that Rett syndrome patients have screaming high levels of OTX2. If we use our little peptide manipulation, RK peptide, into the visual cortex, we can rescue this hyper-connected situation and the hyperfunctional parvalbumin cells in the MECP2 knockout mouse. The cortical thinning, which is characteristic of Rett syndrome mice, is also rescued by knocking out OTX2. And in fact, these manipulations here are done in the choroid plexus, not in the cortex itself. If we look at the phenotypic score, which gradually gets worse with age in the Rett mouse model, it doesn't get worse in these animals. And we've effectively doubled, if not longer, their lifespan by deleting OTX2 from the choroid plexus. That was done in males. This is in the heterozygous female mouse. If you do a genetic manipulation or a viral manipulation that affects OTX2 production in the choroid plexus, the severity of the Rett phenotypic score is kept at a low level in these animals, and their locomotor ability, in fact, is improved. They are less likely to fall off a rotating rod, but a movie is worth 1,000 words. So if you look at these mice, I hope you can predict which one has been rescued by OTX2 and which not. They're both MECP2 deficient, but I think you can tell that this one here is relatively motionless, has this kind of tremor. The other one is happy, its fur is shiny, and it will go on to live as long as we let it to, it seems. And yet, we can't cure everything. I want to make that clear. If you lift up these animals, you will get the characteristic hind limb clasping in both models. So I think we're on the road to something that might be treating at least cortical components or cortical aspects of these mice. And of course, we use monogenic models to help us understand broader idiopathic autism as well. If we could understand a little more about the excitatory inhibitory balance in these patients, regardless of their genetic makeup, we might have a way to fix the circuit instead of the gene. And in doing that, we might be able to go some distance to treat the behavioral symptoms that are involved here. I've only talked about one component of so many things that, in fact, have been studied, all related to the parvalbumin cell. This evening, I'll talk more about this aspect here, which I find fascinating. But I just want to leave you with the notion that, in fact, this lowly mouse visual cortex model has given us new insights, basically two important ones, that excitatory inhibitory balance determines when the critical period will start, and that brake-like factors like the perineuronal net, a good example, come on to actively counteract plasticity beyond a certain age. But with some clever manipulation, the nervous system might be able to lift those brakes when learning is needed. And today I offered one possibility that these pivotal parvalbumin cells, by generating gamma oscillations, might be able to do that. They are also a reservoir for many molecular factors that are interesting plasticity molecules, and if you tamper with those, you might also be able to reset the system. So with that, I'll stop and thank a number of people. In particular, I thank Michela Fagiolini, who has been my lifelong collaborator in this work and has been a pivotal player in translating this to mouse models of autism. Alain Prochance, our brother in arms on the OTX2 story. Kim Do, who is the guru of oxidative stress in schizophrenia. And so many people in the laboratory. I tried to mention them on all the slides. The newest data, Henry Lee, who's worked on the parvalbumin alpha-1 knockout mouse model of elevated gamma. Rebecca Ray, who has moved on to a postdoctoral fellowship with Janet Worker. Katie Quast, who is now with Guoping Feng at MIT. And Delma Kayati, who also contributed to the parvalbumin cell synaptic plasticity. And of course, I thank all the funding agencies in Japan and in the United States who make this work possible, and all of you for your attention. Thank you so much.
So, Takao, is this, I think we're on, okay. So elegant, really beautiful work. We're going to take some questions. I may start with the first one. It's really intriguing, this finding that the gamma oscillations are producing the oxidative stress, which is required to dissolve this perineuronal net. Is that the correct?
That's how we see it, yeah.
That's the way you see it. So what are the, it's intriguing because more and more I've been hearing, there's been a number of reports about oxidative stress as a risk factor for depression and anxiety.
Yeah.
I think it's hard to, and it seems like those reports have been going both ways. There's some that feel like oxidative stress may actually have some beneficial effects, other ones indicating it may be a risk factor.
Right.
Any thoughts about how these could be put together? I mean, these are looking at enzymes like glycosylase and other.
Yeah, so I think there are two things. I think there are degrees of oxidative stress. My hunch is that some oxidative stress is contributing to normal plasticity, and that instability, and we can talk about what the instability is in a minute, is a necessary component of normal critical period plasticity. The nets are not normally there then, or not as strong. And so the system is able to go into this kind of transient state of redox imbalance. If it continues for too long without protection from a net, it becomes pathological, say in Sabina Berretta's case, looking in the schizophrenic postmortem tissue. There may be a reason why that gamma is only induced for a few hours. You don't want to do that for this very reason under normal plasticity conditions. And so that's one point. The second point is where the stress is found. So we focused very much on the parvalbumin cell. They are the hardest working neuron in the cortex. They can fire at several hundred hertz. They're chock full of mitochondria, ripe for generating reactive oxygen species, and in fact are vulnerable in pretty much every mental illness. People have reported something wrong with parvalbumin cells. So, but that's at the end of the day, the final pathological picture. What happens en route to that point is extremely important to learn. And so I think a part of the confusion might be if you homogenize brain and measure redox in a number of cell types, and parvalbumin is only one small component of that, you might not be getting the right picture or conflicting pictures. And so we really need better tools. One line of investigation with Kim, in fact, is looking at fluorescent dynamic redox indicators that we can look at in vivo, in fact. And so I think that this is an exciting time because we've done a broad cataloging of these different models, but now we can go in with cellular precision as well as timing developmentally. Yeah.
That's great. Questions for Dr. Hensch? Bill.
That was wonderful, Takao, but I knew it was going to happen. So I just benefit from hearing your story. It's getting more and more elaborate and just wonderful as the day goes on. I'm wondering, and this is the first time I thought about it hearing your talk, is the opportunity for gamma activity, which occurs naturally in a biphasic way during REM sleep. If that's opportunity for REM to exert its effect is very different whether you're within or without that the critical periods, I would think.
Right, that's a great question. So for a long time, we've been interested in the role of sleep in critical period plasticity. Of course, everyone knows infants are sleeping much more. Why is that? Well, this could be one reason, that it's auto-regulating some of the plasticity machinery. I mentioned the little bursts of replay during REM, a kind of wake-like state as far as the cortex is concerned. And that might be more readily seen in younger perineuronal net less brain. And so definitely this is, now we can parameterize, why is gamma for 3 hours particularly interesting? Does it matter if it's happening during the REM or waking or both? We do know, Marcos Frank's work, our work, that sleep during the critical period is very important for consolidating changes that are triggered by the first few hours of deprivation, say. You don't need continuous 12 hours of visual experience. The first 6 set the stage, and then you sleep. And that seems to be important for executing the downstream rewiring. So, yeah, I'm totally on board with your idea that the sleep activity patterns could become very important. Yeah.
Other questions? Oh, Jeff.
Takao, 3 brief questions. One is OTX2 a druggable target, in your opinion?
Yeah, so we're actually running a screen now for drugs that will up or down regulate OTX2 in the choroid in particular. Thankfully, nature has put it there, and we can address that maybe even from the blood periphery. Yeah.
And with regard to the schizophrenia work, you showed that there's increased gamma activity, which occurs in the prodromal phase, which then progresses to diminished levels, and presumably is related to the alteration in the Parvalbumin interneuron activity. So within the sort of schizophrenia literature, the kind of dogma in terms of etiologic or pathophysiologic hypotheses has been a neurodevelopmental model. Mm-hmm. And which doesn't really afford a role for a progressive or degenerative process, because there's not a history of evidence which support any kind of neuropathologic stigmata of neurodegeneration. But you know, the model you're suggesting suggests that there's the potential for this progression where you have a diminution by some mechanism or another of this inhibitory activity.
Mm-hmm. That's right. So what we're suggesting is there could be something about parvalbumin cells early on that's building up to this moment that reflects oxidative stress. And there was a paper in Neuron by Patricia O'Donnell and Kim Do, you may have seen maybe a year and a half ago, where they do a kind of prophylactic treatment of antioxidants to a variety of animal models that of schizophrenia. And they were able to delay symptoms by giving N-acetylcysteine in this period before. And so that's one very strong implication of the work.
And lastly, you know, hearing your summary of your prior work, you get a newfound respect for the importance of the non-sort of pyramidal cell, the non-excitatory cells in terms of brain development and function. Do you think that a similar thing, 'cause I remember, I'm forgetting who the principal investigator or scientists were in this area, but do you think that glial cells also have been sort of undervalued in the role that they play in terms of shaping development?
Yeah, glia are very interesting, in fact. So, for example, who makes the perineuronal net? In fact, parvalbumin cells must contribute something because they're attracting all of these factors, but it seems they're simply putting out a scaffolding, and a large part of the chondroitin sulfate proteoglycans, all of those things are coming from glial cells. Microglia are also important. We suspect that they're involved in pruning of synapses. I just shown you that the synapses onto these parvalbumin cells are quite plastic. Well, if you don't have a perineuronal net, they're more accessible. The synaptic boutons are more accessible to microglial pruning. And so that might be the physical way in which it prevents this pivotal cell from changing.
Other questions? I have one more. So it's, yeah, maybe it's obvious, I'm just not seeing it, but the fact that if you do the monocular deprivation in the light, obviously you generate this gamma signal. But if you don't do it in the dark, you don't get that signal. That's kind of obvious, I guess. But is that an indication that the remaining eye is somehow involved in generating this signal, or because?
Yeah. So it went by very quickly, but the parvalbumin cells, unlike pyramidal neurons, are really very equally innervated by both eyes. So it's long been a mystery. Why does it take several days to see the ocular dominance plasticity? And that's because people have always been recording the excitatory cells. The inhibitory neurons, in fact, receive equal input from left and right eye. And the instant you patch one, there's still 50% drive from the other one, which can drive the gamma oscillation. And then subsequent stages happen.
If there's not other people, I'm crowding out. I have two other things I'd like to ask. First, you at the end, I don't know if it was because of time or what, you sort of briefly touched on autism. Could you elaborate on your thoughts about, you described in more detail how these mechanisms may be relevant for Rett syndrome and autism, which is a related but different neurodevelopmentalism. Could you just elaborate on your thinking about that?
Right. Well, it comes from the excitatory inhibitory balance ideas around autism, originally articulated by Merzenich and Rubinstein, in part based on work that we were showing that inhibitory neurons develop slower and determine these critical windows of development. EI balance can be disrupted either by having too much inhibition, like I showed in the Rett, or a delayed inhibition, as you see in Fragile X or Shank3 or other kinds of monogenic mouse models. And so we think that this balancing act can go either way, and oddly leads to similar kinds of behavioral outcomes. The treatment, however, would be very different. For Rett syndrome, where we know the inhibition is maturing too fast, we took a strategy to knock it down and slow it down. In other cases where inhibition is maturing too slowly, I'd say probably the more common form of what's going on, we want to find ways to pump up the inhibition. And that's why we're interested in, say, bidirectional control of OTX2, if we could get it increased in those kinds of situations. But what's crucial is knowing the status of the EI balance. And so the visually evoked potential might be a good way to do that. If you can assay quickly some signature of cortical EI imbalance, whether it's too much or too little inhibition, that would then determine a course of treatment to improve the balance.
So I want to sneak in a third thing then. So this may be somewhat naive, but if looking at various brain disorders in the context of development and maturation, and particularly with regard to the critical period concept, you have classic neurodevelopmental disorders, which the phenotype begins to be manifest, almost immediately, or at least during infancy when that stage of development occurs, and autism is a little bit later occurring than maybe some others like Fragile X or Rett. And then you have other clear genetic brain disorders like schizophrenia or Huntington's disease, which manifests later, schizophrenia in adolescence, post-pubertally, Huntington's during middle age for the most part. Would you see those in a similar context?
Well, that's a broad sweep. But with Occam's razor, I would start with EI imbalance and in which direction these imbalances are emerging. We're talking about different brain regions which are developing at different times. And certainly many of the players I mentioned today are appearing in these other disorders or diseases. As I'll talk about tonight briefly, a consequence of not closing a critical period might be degeneration or vulnerability to degeneration. And so excessive plasticity is not necessarily a good thing. And so these kinds of the balancing act, as I keep talking, can be focused on different conditions, as you were mentioning.
Well, if you thought that was a broad sweep, this is going to be even broader. This is it seems to us psychiatrists that the brain is a singularity, or at least exceptional from any other organ, which really has a very clear purpose-driven function. Whether it's the heart, the kidneys, the GI system, the liver. The brain is so multifaceted in its functions, and also comparably complex in its anatomy and I guess sort of its organizing principles. And you're demonstrating this with your exquisite work, at the cellular molecular level in terms of development. I guess the question is there? It's sort of like the theoretical physics. Is there like a grand sort of theory? I mean, do you speculate, even though your work is focused, as to what sort of a broader organizing principle or mechanism is, since there's obviously so many different aspects of brain physiology occurring concurrently during periods of development that are influenced by genes as well as environment. Do you, in your sort of less focused moments, sort of think about this more broadly or grandiosely?
Yeah. So, what we've worked on is a kind of common idea across systems at probably the first level of innervation to the cortex. Long-range projections are sculpted in development through experience by this local cortical excitatory inhibitory balance. And this theme seems to reiterate when you move from visual to auditory to other parts of cortex. And so from a cellular point of view, I think we are coming up with a kind of developmental model of how local circuits influence long-range connectivities. And, this is my just speculative slide. So this could be dorsomedial thalamus, it could be amygdala, it could be, different aspects. But of course, there are later stages of plasticity, and there are certain brain regions, especially in humans, that are less well endowed in brake-like factors like perineuronal nets, and they stay plastic throughout life. And that's part of what I was alluding to before. Those are the regions that go out first in Alzheimer's degeneration. And so there may be a price to pay. But I do think there is probably some kind of core principle that we could start to imagine. And, it's very early days to say much more than that.
Oh, the last thing I'll add is that it seems to me, though with such sort of complexity and time sensitivity in this, sort of grand symphonic process that's orchestrated, there must be a lot of resilience and redundancy that's built in to avoid what would seem to be so many chances for things going wrong.
Yeah, that's right. So there are several brake-like factors. Why do we have so many? And one big project is to understand what happens if you remove the perineuronal net to the links molecule, or vice versa. Why is there so much redundancy in function to dampen plasticity? That's one area of interest.
Well, I think we should stop there because we need to take a few photos and then get you up to the resident lunch at 12:30.
Okay. All right.
But let's thank Takao again and congratulate him.